So my name is Gabriele, and I'm working in a genetic testing lab. I'm a geneticist, but I also trained in dermatology, and I was doing research on keratin and also inherited skin disorders for quite some years. So I want to take you on a journey into the world of keratins that goes a little deeper, but I'm very grateful for the introductions I had from Peter and from Francis, who explained already about keratin, so a lot will come back, but that might help you to really understand how those disorders occur. So what we want to talk about today is what are keratins, where can we find them, what happens in keratin disorders, and which keratin disorders are there. So let's dive just in and start answering the questions, what are keratins? And we heard before, there are structural proteins, there are fibrous and very insoluble structures inside the cells. Humans are not the only ones who use keratins extensively. They are also found in other mammals, in reptiles or birds and amphibians. And they can form extremely strong structures such as the hoofs, the nails, the claws. And in other species they are very soft, for example the wool of sheep or the feathers on birds. They are all produced by keratins. They are the primary components of what's called epithelial cells. These are the cells that form the barrier to our environment. So they help to protect us mechanically. For example, that's very important for the skin or the lining inside the lungs or the lining in our guts. They help us to protect us from loss of water and other nutrients from our body into the environment and vice versa. They protect us from sun damage, for example, the hair. They might be used as a defense mechanism if you think about your nails. And inside the cells, there are very important scaffold for other proteins along so they can travel or assemble. There are many different keratins. As Francis has said, with sequencing the human genome, we found that there are 54 distinct genes which produce different types of keratins. Based on their chemical properties, they are split into type 1 keratins, that's shown down here, and type 2 keratins. And those genes for these types are clustered on two different chromosomes, on the long arm of chromosome 12 and 17. What's shown here are the keratins that are expressed in the skin, hair, nails, and other epithelial cells. But in addition to that, there is an equally number of proteins that are found in hair and hair follicles, so the number of genes is even larger than that. Now, Peter and Francis already talked a little bit about the structure of keratin, so a lot will be just a repetition of what you heard before. Keratins are made out of sequence of amino acids, and those amino acids can assume a secondary structure. They fold onto each other. And the most important region of a keratin is the central, what's called rod domain, where the amino acids fold a helix that is very tightly wound. And this structure is extremely stable and inflexible. And therefore, it's interrupted here at those little regions that are called linkers to be more flexible so the protein can actually bend. This central domain is extremely conserved across different species, so the composition of amino acids is going to be the same, and also between individuals, each one of us, the sequence will not differ. And this is particularly true at the beginning and end of this central domain, which is very important for the function of the protein, as we will explain later on. The head and tail domains are more variable between individuals and between different species. Each one of those domains fulfills specific functions and has particular importance. And as Francis pointed out, the beginning and the end domains are the ones that are crucial for the alignment and assembly of keratin filaments. So keratin proteins are aggregate together into what's called keratin intermediate filaments that you see here. So if you look at this under the electron microscope, when you have a really high magnification of 100,000-fold, then you can see that individual because that pointer is somewhere. Individual filaments, as in the top left corner, forms those rod-like structures. So structures usually are not present just individually, but they collapse into each other and form bundles at the bottom right-hand corner. And in the middle you see how those filaments are forming inside the cells, between two skin cells, two keratinocytes. So they stretch from one side of the cell to another, and mesh the nucleus so to keep it in place, and they also connect at the points of cell-cell contact to another cell with the keratin bundles of the neighboring cell, forming really a network all across our skin. Now it's actually very interesting how those filaments are formed themselves. So there are individual keratin proteins. Oh, is this? No, you can't hear me. Is that working out? If I do like this, I want to be sure to.
Yeah.
Okay, can you see? So these are individual proteins of one type of keratin, but they don't like to be alone and degrade immediately unless they can find a partner keratin from the other type. So you always will have a type 1 keratin assembly with a type 2 keratin. You can imagine it's like if you take your arms and pull them on top of each other, and you have your hands sticking out, which are the variable regions, and here you have the very stable rod domain in the centers that are twisted around each other. Those dimers, which is the name for two molecules being aligned together, snap together with others and are staggered on top. So they form tetramers, and then they can produce sheets of those elements where they get longer and longer and longer and just tack on. And in the end, they're basically rolling together into this 10 millimeter diameter worm-like filament structure. If this is all too complicated for you, we can make it really much easier to remember. Just think about the days when you played with Lego bricks, and the Lego bricks were still just simple bricks and not all kinds of fancy things and batteries and whatever. So you can stack multiple together with some overlap, and it gives a very stable structure that can, as in this example here, form an enormously stable construct. This is a tower built out of 550,000 Lego bricks. It's standing in Munich in Germany, and it is 98.3 feet tall. So keratins can be really strong and stable. They are expressed in many different tissues, and here are some examples where you can find them. We talked about the skin, the hair, and nails, and on palms and soles, but also inside the mouth, on the tongue, in the esophagus, in the GI tract, in some simple epithelia, like in the liver, or the outside surface of our eyes. The transparent cornea also is made by keratins. Each different tissue has different types of keratins that are helping to fulfill the function in that particular tissue. So, as I explained before, keratins are usually coming in pairs. So you have pairs that are expressed in the skin. You have keratins 3 and 12 that are found in the cornea, 4 and 13 in the mouth and other urogenital epithelias, keratin 6, 6A, 6B, and 17 that are resulting in Pachyonychia Congenita are found in the skin and hair follicles and others, and we go into that. So as you see, there is a pattern to it, but it's important to remember that they always come in pairs. And as Peter explained, our skin is really a renewing organ. So we have at the bottom the basal cells that regenerate themselves and produce more cells. They move upward, and as they go through a process of differentiation and changes to form the outer layers, the dead skin cells, they express different types of keratins. So in basal cells, you find particularly keratin 5 and 14, and some others like 6B and 17. But in the more differentiated cells, you find keratin 1 and 10 and 9, or 6A and 16, and so forth. So that's very important to remember. If you Francis provided me with this beautiful slide where you can see this by staining skin sections with different keratin antibodies. So here you see the basal layer lighting up with those green staining for keratin 5, and in the other layers of the skin sparing the basal layer is keratin 10, and more prominent on the top, most differentiated layers, you can find keratin 2. Now, the PC genes are found not only in the epidermis, as we have seen right now for the others, but more widespread in other tissues. So in hair follicles, in the adjacent sebaceous glands, in sweat glands, in the nail bed and nail fold, in the mouth and throat, and also in the, apparently in the tooth spots during the embryonic development of the teeth is where you find keratin 17. I couldn't find a good picture to show how the keratins are distributed in human skin, so here are the same sections through skin of the tongue, through the sections of the tongue as well as inside the mouth, the different epithelias, and I don't know if you can see it with the lights being up so much, but the red stain is always one type of keratin, keratin 13, which you can see is lighting up the entire epithelium in the tongue as well as inside the mouth. And then the green is here in this picture, keratin 14, which again is very basal. In this picture, green represents keratin 6, which highlights up clusters of cells inside the tongue epithelium, but mainly the papillae where you taste things. And keratin 6 is found in little clusters. And this is the most adorable picture I've ever come across when it comes to keratins. This is from Pierre Colombe's lab. It's a beautiful work where they made transgenic mice, genetically altered mice, where they basically just tag keratin 17 with a green fluorescent protein. And these are live images of an embryo during development. And you can see everywhere you find keratin 17, it gives the green fluorescent glow. So this is all the hair follicles are lighting up and the whiskers of the mouse. So I named it Charlie. I think it's the cutest, most adorable thing I've seen. And then they did the same studies looking more closely under the microscope, so you can see it on the tongue and the papillae on the tongue, as well as here where you see the nail plate and the tip of the digits and the nail bed and nail folds here expressing keratin 17. So it's not moving forward. This is an issue. So what if something goes wrong with those keratins and the way they are formed? Then we have obviously a keratin disorder. Let's go back to this slide that we showed so many times before and remember what Francis illustrated to us, how keratin mutations can occur by being just a spelling error affecting one single base in that protein and changing it on one position. Now those mutations, as she explained, really cluster in those regions, and those bars represent the frequency with which we see mutations in many different keratins. And clearly there are hotspots and clustering of those mutations, and that's why we usually start first to look for mutations in those areas. If you have a mutation at those overlapping ends of that central domain, it really changes how those filaments can assemble to each other. It's almost like if you take a Lego or a Duplo stone and then you try to build another one on top, but it's not a perfect match. It's a different brand. Let's say you have Lego and Mega and they don't match up. And if you have just one stone that doesn't match, then your structure might still be okay and fine. But the more you have those unmatching pieces that don't fit together, it's not tight, then your whole structure crumbles and falls apart. So that's also happening with the filaments, and as you can see on the right-hand side where we have a mutation that the filaments are not long and nice and elongated, but they are short and fragmented if you grow them inside a petri dish. And in human cells, as you can see here, they clump and they are unable to form the nice filaments that we see under normal conditions, which results in fragility of those cells. They collapse and they can burst and blisters can occur. And this is what's causing really all kinds of different keratin disorders. And if you remember this picture from before, depending on which keratin gene is expressed in a layer of the cells or of the epidermis in this case, you will find a different disorder that has different clinical presentations depending where you need the keratins in your body. This is a long list of all the 22 different keratin disorders reported before, and obviously you neither need to read it, nor can I in this short amount of time go over each one of those. So I want to highlight just very few. One is Epidermolysis Bullosa Simplex, which is a blistering skin disorder that is more common than Pachyonychia Congenita, and the blistering occurs at the bottom cells of the skin. And depending on where you find those blisters and how common they are, there are 4 different clinical types that can be distinguished. But the main feature is that the cells in the basal layer of the skin are not as stable. They can collapse under friction and pressure, and then that results in blisters. As you can see here under the microscope, this is the skin rupture right here, and then clinically how it presents in an individual. And here are more pictures. Those blisters, like in PC, affect the hands and the soles of the feet because there is more mechanical pressures, but they can be anywhere. Another disorder that does have blistering and skin fragility, but more prominently thickening of the skin, is epidermolytic ichthyosis, or also formerly known as epidermolytic hyperkeratosis, or EHK, due to keratin mutations in genes that are expressed in the upper layers of the skin. So it starts out at birth with a blistering disorder almost identical or very reminiscent of EBS, but then later on the skin gets very thick in an overreaction and basically an attempt of the body to produce more cells to be more stable. So here are again some pictures of the inside of the skin under the microscope, where you see the holes punched inside those cells, where you see the white. These are holes where the cells fall apart, which then results in the skin fragility as shown in the picture on your left, and the thickening of the skin following that on the right. And again, just several pictures of the rigid hyperkeratosis that you see in the axillary, or here's the velvety-like surface of the thickening of the skin, and sometimes you can still see blisters even if individuals grow older. Depending on where your keratin is expressed, you might have different clinical features. For example, keratin 10 is not typically found in the skin of palms and soles, so also this patient has very severe hyperkeratosis, the palm is spared, in contrast to individuals with a keratin 1 mutation that is strongly expressed on palms and soles, and therefore this site is more affected. Similar to keratin 1 mutation is also the expression pattern of keratin 9, so you see keratin 9 on palms and soles. But since this keratin is not found anywhere else on the body, you don't have any other locations that are affected, so only the skin of palms and soles is affected. And of course, you heard already a lot about Pachyonychia Congenita, and obviously you are the ones who know the most about it, so I can fly through this and just really, you are familiar with all the pictures and the clinical features. But it's interesting that each one of the keratin genes has its own expression pattern, being basal or in different tissues, and that really then lines up very nicely with the clinical symptoms you have and the features you see in your skin, reflecting where the keratins are expressed. And then the cysts in Pachyonychia Congenita are also very interesting because they're sitting deep in the dermis. Here's the outer layer of our skin with the dermis, and then you see those fluid-filled cysts that are originate from those foamy cells down here, from the sebaceous cells usually associated with hair follicles. And it's basically a similar attempt as on the skin, that you have an overproduction of cells trying to compensate for the fragility that results in those cysts. Very similar to Pachyonychia with the findings inside the mouse, the hyperkeratotic thickened plaques is a disorder, keratin disorder called white sponge nevus, where you see those plaques inside the mouth and on the tongue, which is very reminiscent of what you see in Pachyonychia congenita. But individuals with this disease have two different keratins affected. The expression pattern is different because you also find those keratins in the esophagus and in the urogenital and anal regions of the body. So as individuals with this disease have no involvement of pons and soles or hair follicles, but in those regions. And then you can have keratins that are mutated and affected in the hairs, and then that might result in particular hair shaft abnormalities and disorders, such as in monolatrix that's shown here, where the hair has those regions, periodic regions of thinning of the hair shaft, which looks like beads on a string. So it's called monolatrix for that region, and they also might have alopecia because the hair shafts are fragile and the hair is breaking off. So there are many different keratin disorders that might affect any tissue that expresses keratins. For any of those, and for any other genetic disorder, I think it is important to know what gene and mutation you have. And these are some of the reasons that I think why it's important, and very glad that every one of you knows what their gene and mutation is, because it helps us to identify in the family who is at risk and how is it inherited, and to provide counseling, to have an early diagnosis, to understand the disorder better by understanding the correlations between having a mutation and what are the symptoms for that particular mutation or gene, by understanding better the biology, what I talked about, what keratins do and why we need them, and also to develop tailored therapies and eventually the cure, we need to understand and have this knowledge. Thank you so much for your attention, and